Hello, welcome to PG Pathshala. I'm Dr. Subhradeep Karmakar. I'm an assistant professor in the Department of Biochemistry at All India Institute of Medical Sciences. Today we are going to talk about nucleic acid as a genetic material in prokaryotes and eukaryotes. We are going to see how important the nucleic acids are. So before we go into the topic, let me just bring up, have you ever wondered why there is life predominantly only in the earth? Even though NASA has actually been searching for life elsewhere, but still predominantly life is some kind of thing which you predominantly see on this earth. And the reason for that is nucleic acids. The nucleic acids actually forms the part of what you call as the DNA and the RNA, and these are the genetic material, and these are the units for hereditary. So they actually helps in the transfer of the genetic informations from generation to generations. And if that nucleic acid has been discovered anywhere in this planet, then you could safely say that there's a chances of life been thriving over there. So back in 1989, Frederick Michers isolated an acidic compound from the salmon sperms and actually given them a name as nucleic acids, or nuclein. Actually, at the time he told us nuclein, and it's now called the nucleic acid. Now the nucleic acids are actually the molecules which store the genetic informations, and that genetic information is actually been transcribed in the form of RNA, which is known as transcriptions, and eventually the RNA have been translated to form the proteins, which we call as translation. So transcriptions and translations actually is the two predominant way by which the information which has been present in the nucleic acid is actually been transferred, and the cell actually can understand that message, and that is important for the function of the cells or for all the cellular functions. Now coming back to a brief history. So back in 1869, as I just mentioned, Michel actually isolated nuclein from soiled bandages. I mean, there's bandages from the wounds and all, and he actually isolated the nuclein from there. And back in 1902, Garrel actually studied some rare genetic disorders like alkaptonuria and concluded that this particular gene, there is a specific gene which is actually been defective and has been associated with the alkaptonuria, and eventually that gene actually codes for one protein, and that protein is actually deficient in alkaptonuria. In 1903, Sutton actually talked about the chromosomal structures. In 1913, Morgan actually talked about the gene mapping. In 1926, Summer purified urease and identified the enzyme to be in proteins. Proteins actually coded by the nucleic acid by the process. Back in 1928, Griffith's transforming principle is a chemical transferred from the dead bacteria to the living cells actually caused genetic converted states and therefore converted stains, and therefore this is known as transformations. And again, this is because of the nucleic acids can actually be transferred from one organism to another. In 1944, Avery and MacLeod actually identifies the Griffiths transformation thing, which you just mentioned about there, and they found that that's going to be the nucleic acids. Back in 1947, Shargubs actually talked about the nucleic acids being composed of nucleotides and the base pairing of those nucleotides. And in 1950, Franklin actually discovered the extra structure of the DNA. And back in 1953, everyone knows the world famous experiments of Watson and Crick, where they first proposed that nucleic acid is a double-stranded structures. And once you know the double-stranded structures, we are actually pretty sure that each of these individual strands is actually complementary to each other. So what does that mean? That one strand can actually help us to know the information of the other strand, and this is very important because during the time of cell divisions, one of the strands actually acts as a template and actually helps in the formation of the other strands. The nucleic acid is actually been composed of what we call as the nitrogenous bases, which is known as of two different types, purines and pyrimidines, and they are actually been present in a way that the purines actually combines with the pyrimidines. Now there is a definite way in which they actually combine. So there is an adenine which actually combines with thymine, and there is a cytosine that actually been base pairs with cytosine with guanosine. Sorry. So we have the AT and the GC base pairing. So one of them is A and T double bonds and G and C triple bonds. Now, we cannot have different combinations because of the sizes are different, and therefore they are going to have steric hindrances, and therefore that is the only one efficient way in which A has to pair with T and C has to pair with G. Now, in addition to these base pairs, nucleic acid also has sugars. Depending upon what kind of sugar it is, whether it is an deoxy sugar or an oxy sugar, it's going to be either an RNA or an DNA. So DNA has a deoxy sugar. In addition to that, they are also the phosphate backbone, and this phosphate backbone is what that actually gives the nucleic acids the negative charge. So if you say nucleic acids like the DNA or the RNA, they are all negative in charge, and therefore they move or migrate in the positive direction during the process known as electrophoresis. And this negative charge of the nucleic acid is predominantly because it contains the phosphate backbone, which actually gives them a negative charge. And again, we are going to see later in different modules that how this negative charged backbone of the DNA or the RNA or the nucleic acids act as a scaffold which actually attracts the positively charged proteins known as histones, and that actually helps in the packaging of the nucleic acids. And this efficient packaging of the nucleic acid is very important, so that in the small tiny space within the nuclei, the entire DNA, which is enormously long, can be packaged and been stored over there. Now there is the two nucleic acids, the repeating units are known as the nucleotides, and these nucleotides actually joined, as I mentioned, by something called the phosphodiester linkages. And that's the reason why you also say that the nucleic acid is a polymer. So small monomeric units, they've been repeated n number of times to form the polymers, and this, so they are actually being joined by this phosphodiester linkage, and that's very important. Now another feature of the nucleic acids is that they can act as a genetic material or material for transmission of the genetic informations from generation to generations, and this we just mentioned is because of the single stranded, the capacity of each of the strands to act as a template and give the information for the synthesis of these other strands. Now there are different types of the bases which is actually been present. So bases are the nitrogenous bases which I just mentioned, and those bases are actually the purines and the pyrimidines. So among the nucleic acids, as I mentioned, there are two types of nucleic acids. One is deoxyribonucleic acids, depending upon whether it's a deoxy sugar, and there's a ribonucleic acid depending upon whether there's an oxy sugar. So we call as in DNA and we call as an RNA. Now essentially RNA is the one that actually transfers the information which has been present in the DNA, and actually been those RNA molecules eventually comes out of the nuclei and sits in the ribosomes and produces the proteins and encodes the proteins. So this is the way in which the information is actually transferred right from the DNA to that of the RNA through the RNA to form the proteins, because DNA is so large, just can't come out of the nucleus. In addition to that, one very interesting thing about nucleic acid is that these two species of the nucleic acids, the DNA and the RNA, they have different stability. So just remember the DNA is very stable, RNA is very unstable. And the opposite is about another thing, that is the enzyme that actually degrades this nucleic acid. So DNase is the enzyme that actually degrades DNA, and RNase is the enzyme that actually degrades RNA. So there is an interesting flip over here. So DNA is very stable, but the DNase enzyme is very unstable. RNA is very unstable, but the RNase enzyme is very stable. So there is an opposite reciprocal relationship, and possibly this is because the DNA is actually genetic material which has the responsibility of transferring the and carrying the information, hereditary information from generation to generation. And that is the reason why it is a predominant molecule which has to be preserved at any cost. That is the reason possibly the DNA is so unstable, because it should not survive too long, else there is a chance that it might degrade the DNA. On the other hand, the RNA is a transient molecule, it actually carries the information of the DNA, and then eventually you don't want RNA to stay too longer, because it's a transient molecule that carries the information and then that's it. We don't want new informations. Now if imagine if the RNA is going to survive too long, of course there are some RNA which actually has a longer half-life, but even then they are less than that of the DNA. And if the RNA is going to survive too long, suppose, just imagine, then what will happen is that particular information will no longer be transient, but rather it is going to stay forever, and that is going to create a problem in the cellular physiology. We don't want that, and that is the reason why the RNA degrading enzyme is very stable, because it makes sure that the RNA is not been preserved for too long. Now, this is this two dichotomy over there. Another very important thing about DNA and RNA is that mostly we are aware of that the DNA is double stranded and the RNA is actually single stranded. In addition to that, it is also very important that among the RNA there are actually different classes of RNA. Okay, so there is mRNA, which is actually known as the messenger RNA, that actually transfers the genetic information from the DNA and encodes the proteins. And then there is a ribosomal RNA, which is the part of the ribosomes. And then there is the transfer RNAs, or the tRNAs, and those tRNAs are actually responsible for carrying the different amino acids. And then take those amino acids actually been attached together to form a protein. Now during a module on protein translation, someone is going to talk about this in greater detail. Now about this DNA and RNA, another important thing what we should remember is that, look, this DNA is a genetic material, and this genetic material is being packaged within the cell in a very small tiny volume. I just mentioned initially that because of the negative charge of the DNA, because of this phosphate residues, it can interact with the proteins which are positive in charge. Those proteins are known as histone proteins and other kind of proteins, and together they forms a very tightly packed structure. That is very important for the DNA to be stored within the nucleus. But that actually solves the problem of space, because if DNA is so large, you cannot keep the DNA just like that. You cannot store it in a small tiny nucleus. But as a result of this packaging of the DNA, it actually introduces another problem. And what is the problem is that the information in the DNA is cryptic, it's been hidden. It is actually been kind of condensed in a very small tiny space. Now, how to read that information? That information is actually read by a process when there is a local melting of the DNA. And as a result of this local melting of the DNA, transiently that information is actually read. And that is actually a very important part of what we're going to talk in subsequent modules known as the expression of the gene expressions. And as a result of this process known as gene expression, tiny portions of the DNA or the locuses of the DNA, specific regions of the DNA are actually being opened up, and they actually get accessible to the different proteins and other factors which actually can read that particular information. And that particular DNA which has been completely condensed is known as the heterochromatin or an inactive chromatin. And that particular region of the DNA that actually opens up and gets accessible to different proteins that can actually interact with this is known as the euchromatins or the active chromatin. Remember that the euchromatins are the one that actually encodes or that actually helps, that actually initiates the process of the transcriptions when the DNA actually forms RNA. But the heterochromatin is the silent region, inactive regions, and that actually regions essentially is almost inactive. So to summarize the today's module, we can actually have a few very important points. Number one is that DNA is a double-stranded molecule. It actually stands for deoxyribonucleic acids, and RNA is a single-stranded molecule that is known as ribonucleic acids. And remember, this DNA and the RNA both can actually act as a genetic material. There are viruses which are known as RNA viruses. So there the RNA is the source of the genetic material. For us in humans and eukaryotes, mostly it is the DNA. Now, in addition to that, we also seen that the DNA, because of the double-stranded nature, it has actually purines and the pyrimidines which have been joined together, and there is a definite way in which the nucleotides actually been nitrogenous bases actually forms a link with each other. So adenine always pairs with thymine, and cytosine always pairs with the guanosines. So it is an AT and GC linkages. DNA structure has actually been solved by the Watson and Crick, and that's a fantastic discovery because once they solved the structure, they found that the two strands are actually complementary. In other words, one strand of the DNA is actually been sufficient to give the information about the other strand, and therefore this is very helpful in because of the reason that during the time of cell division, when one of the strand actually comes from one of the parents, then that particular strand actually can have all the informations to encode the other strands. Now also we have seen that DNA is pretty large molecules, therefore there is a question of packaging of the DNA, and this is essentially been done because of the reason that DNA is actually been negative in charge and it can actually interact with the proteins known as histones which are positive in charge, and together they actually forms a tight structure, and that tight structure has actually been helped in the packaging of the DNA. And we also learned that this unpackaging of the DNA, the local unwinding of the DNA is actually impossible to read the information of the DNA, and all of this actually been critical for DNA to function as a genetic material and as a unit for hereditary, because this information of the DNA is been not restricted within itself. It gets transferred into an RNA molecule known as the process of transcriptions, and from the RNA it actually goes out of the nuclei and sits on the ribosomes and forms the proteins which is known as the translations. Thank you.